Homeostasis of oncogenes. A presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. We read in Cell, cancer results from alteration in critical regulatory genes that control cell proliferation and differentiation and survival. Studies of tumor viruses revealed that specific genes, called oncogenes, are capable of inducing cell transformation, thereby providing the first highlight or insight into the molecular basis of cancer. It's very interesting, but let me change a little the introduction. Genetic cancer results from alteration of critical regulatory genes. Because we are talking about cancer, which is a disease of the human organism. So while we humans may carry all kinds of oncogenes and remain healthy, since disease is a manifestation of a complex system. So let's talk about the two interpretations of homeostasis. This is the first interpretation, that all the processes in the body maintain homeostasis, but we are interested only in the upper part of this homeostasis, the homeostasis of the human body, which we then discuss in this representation of homeostasis, which is called also milieu intérieur. So homeostasis is a self-regulating complex system by which organism maintains stability and maximal optimality. So we are talking about this proto-oncogene. It is a normal gene that could become an oncogene due to mutation or incorrect expression. And oncogene is a gene that has a potential to cause cancer, which we know it's a molecular cancer. So let's look at the representation of the two states. Here we have homeostasis of proto-oncogene, and here state 2 is a homeostasis of oncogene. What does it mean? That we look at these proto-oncogene and oncogene, but they interact with all processes or many processes in the organism. So, and we know that despite the interaction, proto-oncogene proceeds to become an oncogene. Now we ask, is it possible to reverse this relationship, that oncogene be transformed into a proto-oncogene? And we know that it's impossible, at least from the theory which we are discussing, despite many processes which are involved. But we have a solution which is provided by the homeostasis. All is needed to delay the exit from state 1 to state 2, which means the proto-oncogene state delay its exit into the second state, which is the oncogene, and we get some influence on reducing the molecular cancer, and etc., etc. This shows you that despite the theory which we are discussing, there are processes which are capable to influence and affect the protogene and oncogene, and in this example we succeed theoretically to promote processes that delay action from proto-oncogene to oncogene state. So conclusion, complex system may modify the oncogene effect. Remember this, more to come.